All right, so this set of notes will be the last 2 pages of your notes. You'll see that we are working on the Hershey-Chase experiment, or the blender experiment, which is the last of the 3 experiments that you'll be learning about in this history notes. So experiment 3, the who, where, when, and purpose. The scientists were named Alfred Hershey and Martha Chase. They were conducting their research in 1952 at the world-renowned research lab called the Cold Spring Harbor Lab in New York. They wanted to conduct experiments to determine if DNA was the inherited material of all cell types. And what's really cool about their research is they used a special type of virus called bacteriophages. Let's learn a little bit more about bacteriophages before we talk about their experiment. So bacteriophages are really cool. They are a type of virus, which again, viruses are non-living. We might talk about that later, but just know that viruses are not living organisms. But viruses, specifically the type that parasitize bacteria, are called bacteriophages. And bacteriophages are made of only 2 molecules. They're made of proteins and DNA. And what's fascinating is that in order for viruses to make more viruses, in order for them to reproduce, they need a host cell from a living organism. So what these bacteriophages do, so I'll look at this picture here. So bacteriophages, a virus, will attach itself to a host cell. And in this case, this is a bacterial cell. And they will inject their genetic material into the host cell. That genetic material will recombine with the host cell's genetic material. And then this host cell will turn into a virus production machine or factory. It basically just pumps out more and more and more and more viruses. Here's a really cool electron microscopy image of this process actually happening. These are all bacteriophages infecting a host cell. Now what's really cool about this is that this doesn't just happen with bacteria. This is exactly how viruses infect our cells too. So imagine you inhale a coronavirus particle, and that coronavirus will attach itself to the respiratory cells in your body, so the epithelial cells of your nose and of your throat and of your lungs. And those coronaviruses will inject their genetic material into your cells, and then your cells will start pumping out more and more viruses. So every time you cough or sneeze, more of these will come out your nose and your mouth. Cool, but not cool at the same time. So with that background knowledge about bacteriophages and how they work, Hershey and Chase reasoned that if you analyze the bacterial cell that was infected with the genetic molecule from the bacteriophage virus, you will therefore find inside of that bacterial cell either protein or DNA from that virus inside of it. So therefore, this would prove which of the 2 molecules is the genetic molecule of all life, because viruses do this same process with our cells too. So therefore, that molecule would have to work in our cells. All right, so here are some of the basics about their experimental design. We already talked a little bit about how they're using bacteriophages, so you definitely want to mention that in your experimental design box. But here's some more important information. In order to determine the different parts of the bacteriophages, they can't just look inside of a cell and be like, oh, that's DNA. They have to analyze it based on the elements that are present. And so we know that there are 2 elements that are unique between DNA and protein. We've learned that DNA contains phosphorus. Protein does not. So therefore, what they did is they soaked a bunch of these bacteriophages in a liquid full of radioactive phosphorus element. And the DNA of these molecules absorbed that radioactive phosphorus. So we've labeled our DNA with radioactive phosphorus, which basically just makes it easier to detect. And in comparison to DNA and phosphorus, we know that proteins contain sulfur. So sulfur is not found in DNA, it's unique to proteins. And so they did the same thing. They labeled the proteins of these bacteriophages with a radioactive sulfur element. So again, this basically just makes it easier for them to do a chemical analysis at the end of this experiment to determine, is there phosphorus inside of the bacteria or is there sulfur? And therefore, is it protein or is it DNA? Okay. We are going to transition to the drawing notes at this point, and we'll come back to this in a second. So let's review what they did for this first batch of bacteriophages. Alfred Hershey and Martha Chase used radioactive sulfur to label protein molecules. So let's write that right here. Phages labeled with radioactive sulfur. which is found only in proteins. Okay, so let's see what happens. So in this beaker is a mixture of radioactive sulfur, and it's a mixture of the bacteriophages and the bacteria that they will infect. So here you can see that this bacteriophage is injecting its genetic material, and remember that at this point they don't know what this genetic molecule is. They don't know if this is protein or if this is DNA. This is the reason why they're doing this. So this bacteriophage injects its genetic molecule into the bacterial cell, but now what they need to do is they need to separate the bacteriophages from the bacterial cells so that they can analyze what's inside of the bacterial cells. Is it DNA or is it protein? And what's really cool about this is that they used, sorry, I'm getting it to pan. There we go. They used, it's called the blender experiment because they actually used blenders. Martha brought her blender in from home to use it in this experiment. So it's kind of an example of creative thinking. And so by putting all of this in a blender, we can separate the bacteriophages from the cells and therefore analyze. They use a centrifuge, which basically think of that as like spinning it really, really fast to separate the lighter particles, which are the bacteriophages, and all of the cell parts from the bacterial cells are stuck in this pellet. And so when they analyze this, they can determine, well, what elements are found up here, and then what elements are found in the cells themselves. And so what they found, sorry, I'm trying to get it to pan again, there we go. Let's see what they found. In the in the liquid, so right up here, in the liquid, they found radioactive sulfur. Radioactive sulfur, sulfur, there we go, in liquid equals protein. So that means that the protein is in the liquid. And so therefore, that means that protein was not injected into the bacterial cells. So therefore, the protein was not the genetic molecule used by these bacteriophages. And so what they found is that inside of the cell, this was not sulfur. Okay, so now we need to do a second trial. We need to confirm that it is in fact DNA in there and not some other mystery molecule. So that's why they do their second trial. So let's take a look at what they did in the second batch. There we go. Batch 2. All right, so for batch 2, they had phages labeled with radioactive phosphorus. With radioactive phosphorus, which is unique to DNA. Proteins do not have it. All right, and so they have this beaker full of radioactive phosphorus elements, which is then absorbed by the bacteriophages. And then they again use that blender to separate the bacteriophages from the bacterial cells so that way they can analyze what was injected into the bacterial cells. And what they found, in fact, was that the radioactive phosphorus was in the cells. So inside of these cells, they had the radioactive phosphorus embedded in their DNA. So let's label that radioactive phosphorus inside the cells equals DNA. So this experiment confirmed that bacteriophages, which are a type of virus, infect their host cell with DNA. And so with this reasoning, well, bacteriophages are viruses that infect bacterial cells, but viruses do the exact same thing to our cells. So therefore, viruses use DNA as their genetic material, and therefore all life uses DNA as the genetic material. And even though viruses again are non-living, because of the fact that they use living host cells to inject the same DNA molecule, therefore it applies. Okay, so let's do a summary at the bottom. So number 1, radioactive sulfur protein was not inserted into host cells. Okay. In the second trial, they found that radioactive phosphorus, which is found in DNA, was inserted into host cells. And so therefore, the final conclusion, here we go, bacteriophages, which are viruses, use DNA as the genetic molecule. All right. Bam, we just solved the problem. We now know that DNA for sure is the genetic molecule used by viruses and therefore by all life, because viruses infect all living things with their genetic molecule, which is confirmed to be DNA. All right, so if you need help writing down the conclusion and significance, here are some tips. So again, the end results showed that the protein in this picture is different than the one in your notes. I apologize. In the first trial, they realized that the radioactive sulfur was found in the liquid, whereas the pellet, which contained the cell parts, did not contain any sulfur. And so therefore, if the sulfur is up here, the proteins are up here, the protein was not injected into the bacterial cells. In comparison, in their second trial, we found that the radioactive sulfur was injected into the cells. To say sulfur, I meant phosphorus. So the phosphorus was injected into the cells, and so therefore that must mean that was DNA. If you need to write down some of the basic parts of this, you can pause the video and write this down. All right, so that concludes our learning about the 3 foundational experiments of the history behind the discovery of DNA as a molecule. Remember that all of these scientists work together indirectly. They publish their research and then they learn from other researchers before them, and then they expand upon the research of previous scientists, which is pretty cool to think about. Thanks for watching. Let me know if you have questions. Have a great day.